Great. Okay, so hi everyone. As you heard, I'm Nikki CREUX. I'm based at FABI at the University of Pretoria in South Africa. And today I'm going to talk a little bit about sunflower anthesis and the delicate balancing act of climate control. So as we all know, pollination and fertilization require an optimal temperature within the flower. And like any good Goldilocks story, if things get too hot or too cold, it can massively affect pollen quality, quantity, germination, or pollen tube growth, pollen pistil interactions, and gametophyte degeneration. And so plants have developed a number of different ways, or evolved a number of different ways to maintain floral temperature. Some of them more tubular flowers or hanging down flowers produce like a little greenhouse effect to maintain their flower temperature. Upright flowers tend to have their petals angled in a way that forces radiation down to the floral organs. In some of the cooler areas, such as where the Arctic poppies are found, they actually heliotrope following the sun to maintain their core floral temperature. And other flowers will open and close in order to maintain their temperature. And it's not just maintaining temperature for floral traits, but also insects benefit from warmer flowers. So there's been several studies showing that bees prefer warmer flowers and warmer nectar. And this study by Harrop et al. In 2017 actually looked at thermoimaging of a number of different flower species. Several Asteraceae were in the mix. And they actually found that there were thermal patterns that could be observed in the different flowers. And in some instances, parts of the flower were significantly warmer than the others. And they also showed that these patterns could be distinguished by bees, and the bees could actually select flowers based on these patterns for nectar, for rewards. So the question then became, so how do sunflowers modulate their microclimate to ensure pollination and reproduction? So when I was a postdoc at UC Davis in the Harmer lab, my colleague Hagop Atimian did a very nice study looking at how the circadian clock regulates heliotropism in sunflowers. And one of the experiments he did was to track, while the sunflower matures, what does the tracking motion look like? And he found that as the sunflower matures, it starts tracking less to the west each time until eventually it stops facing east. And I'm sure we've all been in a sunflower field and seen the blooms facing east through the day. And so we wondered if this easterly orientation had a biologically relevant, was biologically relevant for the plant. So the first thing we did was we planted sunflowers in pots in the field. And then once they finished tracking, we moved, we turned half of them to face west. And then we measured temperature as well as counted insect visits. And the first thing we noticed was that the east-facing plants were much, much warmer earlier in the day compared to the west-facing plants. And this seemed to coincide with an increase in insect visits early in the morning. So we wondered if the temperature could be having an effect on the insect visits. So then we ran the same experiment. Where we heated a west-facing plant to mimic the temperature changes on the east-facing plant, and then counted insect visits again. And we did see an increase in insect visits on the west-heated plant, suggesting that temperature is helping to coordinate insect visits at specific times. So then I had a question about, well, do these floral temperatures actually change the flower or seed physiology and pollination? And so we went back into our field and we measured insect visits with video analysis again. We did time-lapse photography to determine timing of pollen emergence. We artificially heated our plants, and I harvested florets and dissected them so we could image individual floral organs. And then I measured temperature throughout the experiment. And we also measured some seed traits. So an undergraduate intern in our lab, Sana Saeed, she did most of the measurements for the seed traits. And one of the things we noticed was that the east-facing flowers seem to have larger capitulum diameter, and the seeds width and the seed weight seem to be higher in east-facing plants. Now, this result was locality specific, so we had this observation in Davis, but we didn't actually, our collaborators at Charlottesville and Virginia did not see the same effect. And we suspect solar radiation probably played a role in that, because Davis has a much higher solar radiation during the midsummer months. And this is reflected by early and late planting dates in Davis, where the seed number varies significantly in depending on solar radiation. But there seems to be, at least in Davis, some female fitness advantage to facing east. So then we wondered if there was a male fitness advantage. And so our colleagues at in the Blackman lab did a really nice study, siring experiment study, where they had cytoplasmic male sterile lines in the middle of their field in pots. And then around them, they placed pollen donors with known molecular markers, either facing east or facing west. And then after pollination and seed development, they collected the seed, germinated them, and did molecular screening on the offspring. And even when they changed the pollen donors or did reciprocal crosses and repeated the experiment, in most cases, it appeared that east-facing plants, represented here in red, sired more offspring than west-facing plants. So this made me wonder if there was an interaction with the timing of pollen presentation on the sunflower due to this orientation. So we went back into the field and I photographed east and west heated plants every 15 minutes. And on the photos, for every photo, I counted how many florets had pollen displayed for each photograph across the time series. And what we saw was that early in the morning, both east and west heated plants presented their pollen much earlier in the day compared to the west-facing plants. And that may be a reason why they were siring more offspring. So I wondered how temperature might be regulating this pollen emergence. So we had to think a little bit about how pollen emerges in a sunflower floret. So you have the corolla housing the stigma, and then the anthofilaments and the anthers, which are a fused cylinder around the style and stigma. And once the corolla opens, the anthofilaments elongate, pushing the anther tube out the top of the corolla. And it's inside this tube where the pollen is dehisced. And slowly the style and stigma elongate through the middle of this tube, pushing the pollen out the top, kind of like a plunger mechanism. Then only later or the next day does the stigma open and is receptive to pollen. And I wondered if the elongation of these organs was being affected by temperature. So I dissected florets over a time course, and I measured the length, style, elongation, and stamens. And what we saw was that the style elongation for east and west heated in the red and orange here they had a much faster elongation rate compared to west-facing plants. Interestingly, the anthofilament elongation didn't seem to change and orientation or temperature had very little effect on these. So this suggests that style elongation and anthofilament elongation are regulated separately. To confirm this temperature association that we saw, I did a chamber experiment. So in the chamber, I grew plants and all conditions were kept constant except for temperature. And when I heated one of the plants, the rate of style elongation was increased, and pollen presentation here in the red was also advanced, suggesting that temperature really is regulating style elongation, and pollen emergence. And so after that, I moved on and finished my postdoc, and I relocated back to South Africa, my home, to start my faculty position here at the University of Pretoria. But a lot of these questions were still playing on my mind. So if temperature regulates the timing of pollen emergence and pollinator visits, how would heat stress and heat waves affect the timing of these processes? So many of you saw our MSc student, Uya Mamela, present her MSc results yesterday. And what she did is had all east-facing plants in the field, and she artificially heated a subset of those. And then she measured style elongation, timing of pollen presentation, and insect visits. And in these cases, with the artificially heated plants, both the style elongation and pollen presentation were also advanced, even with higher temperatures than what's naturally seen on east-facing plants. Interestingly, her insect visit counts did not change, and I suspect this is because temperature alone does not regulate floral visitors, and visual cues and light also play an important role, and these plants would look very similar to an insect. While she was doing her study, we were quite lucky to have a heat wave during our summer period. So we also managed to collect some data under natural heat wave and compared this to our normal sunny days. And we found that style elongation was also faster under heat wave conditions, although it seemed that the styles shrink a bit at the end and end up smaller than under normal sunny conditions. The pollen emergence was quite interesting because the pollen didn't emerge much earlier in the day under heat wave conditions, but the rate at which the pollen emerged was much faster. So the time between first floret with pollen and all florets with pollen was very short during heat wave conditions. And under these heat wave conditions, we also noticed that pollinators and insect visits seem to increase at the earlier time points during the collection. And so another experiment that we've got running in the lab is a planting date trial. And this is run by a very capable PhD student, Fraser Mapfumo. And she has 5 planting dates in the field. And a planting date trial is actually quite a powerful tool because if a specific environmental phenomenon hits, you actually have plants of different developmental stages in the field at the same time. And the same heat wave that affected Uya's fields had an influence on this planting date trial. So we had some seeds that had just been planted that month, and so that obviously drastically affected germination, which is to be expected. The plants that did actually germinate were able to recover and actually grew to perform pretty much the same as the earlier planting dates. It's the blue line here. Interestingly, the January planting date, where the plants were about 4 weeks old, really suffered under this short heat wave during February, and the plants never really performed the same as the other planting dates. When we did some floral trait analysis, we noticed that stigma receptivity seems to be increased in these stunted January planting date plants, which raises some interesting questions about adaptation and balancing effects of abiotic stress and increasing plant fitness in a way, if possible. So the next step in our lab, or the next question we started to focus on, is what are the molecular mechanisms regulating floral response to temperature? And I have 2 students who've started working on this, Vera Roda, who's an MSc student, and Ronelle Groenewald, an honors student. And they've started looking at some of the gene families that might be involved in floral traits and abiotic stress, using some of the genomic and transcriptomic databases that Olivia mentioned earlier. And then we're going to start doing some expression studies and gene expression networks to start understanding how these organs are responding to abiotic stresses. And with that, the main take-home messages from the talk are that sunflower orientation affects plant fitness and coordinates pollinator visits. Temperature regulates style elongation and timing of pollen emergence, but not at filament elongation. And the higher temperatures associated with heat waves seem to further increase the rate of style elongation, advance pollen emergence, and insect visits even further. And with that, I need to thank many people, because the work never gets done in isolation. So I need to thank the Harmer Lab, that was my home for many years, and the Blackman Lab, who were incredible support, and together they were a great bunch of friends, and I miss them every day. And then I need to thank the UC Davis and University of Pretoria field management teams, my collaborators here at the South African Agricultural Research Council, Dr. Mali and Dr. Swanefelder, our lab that we have joint lab meetings with, MPPI for being an awesome sounding board, and then the Grain Research Program and the Center of Excellence for Plant Health Biotechnology here at FABI for supporting me as I got my research team going over the last 3 years. And then, of course, we can't leave out the funders, because without them, this work couldn't be done. And thank you for your time.